So this next video is about DNA replication, and we start off with a familiar diagram that you've seen before just so that you know where we are in the cell cycle. So we're in interphase, but we're specifically in the S phase of interphase, and during this time DNA is synthesized. So we're right here in terms of the calendar of the cell's life. So on this diagram, there's quite a bit of review, but there's a few new concepts here. So first of all, you're familiar with the double helix, and you're familiar with the monomer of a double helix being a nucleotide, where you have one sugar, one phosphate, and one nitrogenous base or one nitrogen base. So DNA strands can be said to run what is called anti-parallel to each other. So they're parallel, but running in the opposite directions. And if you look at the sugar molecule, the deoxyribose, on the left side, the oxygens are all facing upward, and on the right side, the oxygens are all facing downwards. So it's kind of like if you had two pens and they're parallel to each other, but the tip and the ends are running in opposite directions. So this way would be parallel, this way would be anti-parallel. And so the strands are in the opposite direction from each other. Remember that adenine always pairs with thymine, and guanine always pairs with cytosine. And there's an important vocabulary word that you guys have been taught in first quarter that was complementary. So the two strands are complementary to each other. So when they open up, it's very easy to build a new complementary strand on the old strand. They're held together in the center by hydrogen bonds, and one sugar, one phosphate, and one nitrogen base. That part would be a nucleotide. Now you see a new thing on here, as well as anti-parallel. Anti-parallel is new to you, but this 3' and 5' designation, that's new. Now what that refers to is the direction of the sugar and which carbon in that sugar ring is at the end. So if you start at the oxygen in the sugar and go clockwise, so this way, and you count the carbons, the first carbon is here, second carbon is here, and third carbon is here, which is why this is labeled the 3' end. If you look here and start at the oxygen and go clockwise, we have the first carbon here, second carbon here, third carbon here, fourth carbon in here, and the fifth carbon is a little harder to see. It's this CH2 that's actually not part of the ring but kind of branched off of the ring, and that is the 5' end. So the next slide actually gets even bigger with that. So we've got our nucleotide, we have our sugar, 1, 2, 3, 4, 5, so this is the fifth carbon, and this would be the 5' end, this would be the 3' end. So it's the fifth carbon in the sugar ring. There's the third carbon in the sugar ring. So remember that when nucleotides go together, water comes out and a larger molecule is built. And the name of that process, you got it, it's dehydration synthesis. So again, remember that we are during the S phase, and the first thing that has to happen before DNA can replicate itself, which means make an exact copy, is it has to open up. But before we get into the details, I want to kind of just review quickly why the DNA has to replicate. Remember that coming up, you're going to be building a new cell, and that new cell is going to have all of the DNA that the parent cell had. It needs that DNA because that DNA is recipes for making any protein that that cell might need to make. And it's sort of obvious that the new cell is going to need its own set of DNA or instructions for making proteins. So first, the double helix is unzipped by an enzyme called DNA helicase. I love that scientists named these enzymes basically based on exactly what they do. So this one opens up the DNA helix. And remember that all enzymes end in ase. Next, an RNA primer tells a new enzyme, DNA polymerase, to begin adding nucleotides. And they can only do it in the 5' to 3' direction on the leading strand. And they build it continuously. Now, just remember when you're thinking about what DNA polymerase does, it builds a polymer of DNA. And then, so it's building a polymer of DNA, and then it has the ase ending. Okay, sorry about that technical difficulty. So, scientists noticed that there were two actual different speeds that this process was going on. That on one side of the DNA replication fork, the new strand was being built rather quickly, and on the other side, slowly. And so they named the side that was being built more quickly the leading strand, and the slower side the lagging strand, because it lagged behind the leading strand. They fairly soon realized the reason why. So the lagging strand is the new strand would be built if it was built continuously in the 3' to 5' direction. But DNA polymerase can't do that. It turns out DNA polymerase can only build in the 5' to 3' direction, adding nucleotides one at a time. So what happens on the lagging strand is that the DNA polymerase must wait until the replication fork is open, jump ahead, and build backwards in the reverse direction. So right here, the first fragment would be here, and it would be built that way, and then the next fragment 5 to 3, and then the next fragment 5 to 3. So they're numbered in the sequence that they are built. This one is the oldest piece, and this one is the newest piece, because this is the area of the replication fork that has been most recently open. Now, these fragments or pieces are referred to as Okazaki fragments, and you can see the spelling right there, and they're named after the scientist who figured out what was going on the lagging strand. So the leading strand is built continuously, and the lagging strand is built discontinuously in pieces. Now, those fragments are joined together by a third enzyme called DNA ligase. And if you're familiar with ligaments being the tissue that holds bones together, you'll realize that this prefix, Lig, that is referring to things being held or bound together. So there are actually a lot of things that can damage DNA and affect the nucleotide sequence in a negative way. You end up with the wrong nucleotide. Certain chemicals can do that. Ultraviolet light can do that, which is why you need to wear sunscreen in a bright sunny day. But there are 15 enzymes that are available to find damaged nucleotides and replace them with the correct nucleotides. So ligase's second job is to seal breaks after repair. Now, unfortunately, the name of the enzymes that find the mistakes and replace the incorrect nucleotides are also called DNA polymerase. So just be careful. So in this slide, it's just sort of a quick review of what goes on the leading strand. First, remember, helicase opens the double helix. And on the leading strand, you have a continuous building of DNA in the same direction that the helicase is moving. And on the lagging strand, it's built 5' to 3', but remember, jumping ahead and building backwards. Now, I want it noted for the record that on the previous slide, the leading strand was at the top and the lagging strand was on the bottom, and this time it's the reverse, because remember, DNA molecules can be in any direction. So you're going to have to be able to look for the 5' and 3' ends to figure out which is the leading and the lagging strand. So remember, when they say that DNA is built in the 5' to 3', they mean the new strand is 5' to 3'. So the template strand that it is building on will be the one with the 3' end open. Because remember that the new strand is going to be built anti-parallel to the original strand. DNA replication occurs in what is considered a semi-conservative method. Now, this diagram on the left shows what scientists thought was one possibility. They thought, okay, so you have this original DNA strand, it opens up, builds a new strand, and the two new strands come together, and the two old strands come back together. The other option that scientists proposed was that the new strand opens up, or the old strands open up, and one new strand is built complementary to one old strand, and they stay together. And it turns out that the option on the right is exactly what happens. So the dark blue represents the original strand, and the light blue represents the new strand, and that is called the semi-conservative model of DNA replication. So this is just another diagram that shows you that each new DNA molecule is built with one old strand and one new strand. What this diagram actually shows you is that when we show you diagrams of DNA replication, we're really only showing you a small piece of what's really going on. Replication actually takes place at many sites on a chromosome all at the same time, and this speeds up replication. So here, what we were looking at, we zoomed in on this one little area, we were looking at was a replication fork, but really it's more like a replication bubble because you have two replication forks that start together in the middle. And you have several replication bubbles that get bigger and bigger until there are no bubbles at all, and you have two new DNA molecules. So students always want to know, well, how fast does this happen? And in prokaryotes, remember those are our bacteria, you get 500 nucleotides added per second. In humans, you get 6 billion bases copied every few hours. So here is a diagram of a replication bubble. And it shows you that you have a leading strand and a lagging strand on each side of the bubble. So again, here is our exit card that you will be going over in class. I don't want to go over the answers right now in this video.